Hello there, ladies and gentlemen. Welcome to another exciting edition of IB Chemistry Flipped Classroom. Possibly even the last edition of IB Chemistry Flipped Classroom, or maybe the second to last. I haven't decided yet. And if you are actually sticking with it and watching this, I appreciate you. Thank you for sticking with me to the very end. The observant among you may notice I gave myself a haircut, which is probably exactly what this haircut looks like, something that somebody gave themselves. But, I'm kind of embracing the baldness, just going short with it, and, who have I got to impress these days? So, this is about taxol, so you should have the student note set that goes along with this. This should be relatively short and sweet here today. Basically, what taxol is being used as a case study or as an example of a very complicated molecule that needs to be produced, and some of the techniques that go along with that. It is an anti-cancer drug. So we talked about enantiomers, and which are non-superimposable mirror images. So this is, again, a case study of a drug that has actually several different chiral type isomers, and you need to make only one because only one works properly. So this is about some of the ways that we can do that. So, as I just mentioned, enantiomers means non-superimposable mirror images, like your hands are non-superimposable mirror images. And in most situations, enantiomers have identical chemical properties, except when interacting with other chiral molecules. Many drugs tend to be fairly complex molecules, so it's pretty often there's going to be one or more chiral centers in there. Remember, to be a chiral center, you need a carbon attached to 4 different things. And because of the way drugs interact with receptors and things like that and the way they work, often only one enantiomer will actually be successful. An analogy here would be like trying to wear a glove on the wrong hand. You can only wear a right-handed glove on your right hand, or you try to put that left-handed glove on there. At the very least, it's going to be kind of weird. So this is another, just example or kind of simplified visual of what you're looking at for enantiomer drug function. So recall that very often drugs work because they're fitting some kind of receptor or some sort of thing on a cell, and they must line up in a very certain way. So we see our enantiomer that is effective. Here's our drug. Here's the binding site, some sort of receptor, and everything lines up nicely. So this is the non-superimposable mirror image of the one we were just looking at, but it doesn't line up. And no matter how you try to rotate it or spin it around, it's never going to line up quite properly. So this drug works. The mirror image version of it doesn't. So, if you just go through normal chemical reaction procedures, you're pretty much going to produce a racemic mixture, or racemate, which means an equal mixture of both enantiomers. So there's some problems with that. It's wasteful, but it's also dangerous, as we spoke about before. But just as an example, the drug ibuprofen is sold as a racemic mixture of 2 enantiomers. They interconvert in the body. Only one is effective, but since they interconvert, neither one is dangerous. They basically just give you both and don't worry about it. We talked about thalidomide. Thalidomide was a form where one drug was effective in producing morning sickness, preventing morning sickness, I should say, for mothers. The other actually caused some pretty horrific birth defects. So nowadays, fortunately, drug manufacturers are very careful about studying drugs and studying enantiomers and how they work. So of all the chiral drugs, it says about 50% of them apparently are given as a single enantiomer. The rest they just give both and don't worry about it. So we're going to be talking about an anti-cancer drug, taxol, here. And taxol is, or I should say cancer is really a thing that is about uncontrolled cell growth. So just a review of how cells grow. Perhaps you remember fondly from your biology days the cell cycle. Cells split into new cells in a process called mitosis. And we don't need to think about the whole cell cycle, but one of the parts is where they line up the DNA lines up in the middle of the cell, the DNA is copied, and then it lines up in the middle of the cell, and this network called the spindle attaches, and basically it grabs onto those chromosomes, pulls them into separate cells, and then the cell splits. So those spindles are called microtubules, and they're made of a protein called tubulin, and basically they're just really helping to pull the cell apart and get everything in the right process. So actively dividing cells are going to be the ones, and rapidly dividing cells that are going to be the ones that are doing this the most. And as we've talked about at several points, if you want a drug, you need something that's going to combat the specific thing that's causing the problem and not harm other things. Again, I've mentioned with cancer, this is tricky because cancer cells are actually your own cells, so it's pretty hard to target them. But if we could target things that are rapidly dividing, taxol is going to affect the most cells that are rapidly dividing, which are cancer cells. It is going to come with some side effects, some significant side effects, but for the most part, it's going to cause the worst problems to the cancerous drugs. So what it does is it binds the tubulin and it prevents the spindle from kind of breaking down, which is normally what happens at kind of the ending latter part of cell division. So then the cell division is going to stop, so hopefully it basically stops the tumor from growing. This was found in a natural source, specific yew trees originally. There's just tiny amounts of it that are present naturally, and you just strip the bark from 200-year-old trees and it kills them. So this was just an environmental disaster and not feasible at all. So they tried to find a way to make taxol a little more effectively. So it has 9 chiral centers, a total of 11. So basically at each one of those 9 chiral centers, there's 2 different possibilities, and the possible combination is actually 2 to the 9th power, which is 512. And out of all of those 512 possibilities, only one works. So it is said to be quote-unquote very chiral, and it's very complicated. So imagine trying to manufacture 512 different ones, or just manufacture randomly and hope the right one popped about, and trying to isolate it would be extremely hard. But you just couldn't do it and manufacture the drug in any quantities. So they do something called asymmetric synthesis, or enantioselective synthesis. Basically means make only the one enantiomer that you want. They do this via something called chiral auxiliaries, and what they do is they force the production of only one enantiomer. And so they attach to a certain spot that they're going to add a piece to, and maybe we want to, there's 2 possibilities. Either the thing is going to attach from this side, or it's going to attach from the other side. We want it to attach from this side, if you can see my hand gestures down there. So basically they put a chiral auxiliary that's going to block whatever piece they're trying to add on the molecule. It's going to block it from being added onto the wrong side, and then they have ways to remove that chiral auxiliary. So they're basically forcing the production of only one enantiomer, which is going to be much less wasteful, and it's going to be easier to make the enantiomer correctly in the first place rather than try to isolate it or separate it. And so we can test to make sure that we have the right enantiomer with a polarimeter. This was always a big struggle, but you can check, remember, for the rotation of plane polarized light with a polarimeter. One enantiomer will rotate plane polarized light in one direction, the other will place it in another direction. So this is a real one here. So we have a light source, the material is placed in here, the thing we're trying to test. Light source is used, which rotates and vibrates in all directions. A non-moving fixed polarimeter changes that light so it's only rotating in one plane. And then as it passes through, if it is a pure sample of one enantiomer, the light will be rotated. So the person here studying will find they would have to twist this little dial right here a certain number of degrees in one direction so that it would match up with that little arrow here. You see how they're not lined up in the right direction? We would twist it this way. The other enantiomer, you'd have to twist it in the opposite direction the same number of degrees. If it was a racemic mixture, it wouldn't rotate the light at all. So that's a way of, once you do this process of testing, you've actually made the right enantiomer. It will rotate the light the proper number of degrees in the right direction in a polarimeter. So taxol is super complicated. If you were going to just make it from scratch, it would take 30 steps, just so hard and so difficult, and you'd have to be using chiral auxiliaries. So what they do is they found something similar to taxol. They call it Tindab, and they get it from the leaves instead of the bark. So you can take the leaves away, and it doesn't kill the trees like the bark does. So you still need 13 steps. That's easier. It's easier than 30, considerably. But at least you're starting from a standpoint where you don't have to start from scratch. It still is pretty inefficient, and people would like to find out new ways to do this. So a couple of things that people are thinking about to make this process greener, a little more environmentally friendly, is maybe you can get some... Typically a really good way to make complex molecules is to somehow trick something that's alive into making it, or take advantage of some sort of living thing that makes it. So there are some fungi that produce taxol in fermentation reactions, so maybe there's a way to do that on an industrial scale and isolate the taxol out of there. And also remember that when living things make substances, they typically make only one enantiomer. All amino acids, for instance, only come in one form. They're made by living things. So if you use living things to make it, you solve that whole enantiomer problem, because the machinery that living things use to make stuff always results in only one enantiomer. So maybe we can get these fungi to do it. Maybe you could, instead of like having to strip the leaves from this thing, maybe you could somehow kind of grow some leaf cells and culture, in like a petri dish, if you will, so you don't have to take leaves off of full-grown natural trees. And again, maybe that could be extended to an industrial scale. Maybe, and I don't know if anyone's thinking about this, but I'm sure they've at least considered the possibility of some genetic engineering. Maybe you could insert genes to make taxol into some kind of bacteria, get it to make a bunch of taxol. They do that with certain drugs. I don't know if they can do it with taxol, but anyway, that's it, folks. Thank you for watching and learning about taxol. Again, if you're watching this, I appreciate it. And we'll hopefully see you again soon. Bye-bye.